Question number 26 for exam 4.1. In general, acid catalysis must be protonated. The active site residue. Which amino acid is likely to participate in acid-base catalysis would be glutamic acid. Aspartate, asparagine is unlikely to participate. Well, we've got cysteine or asparagine. Well, asparagine is the least likely. 27 is C, as is 26. Because asparagine is unlikely to be protonated. Cysteine is also less likely than histidine or aspartic acid or glutamic acid, but it's a better candidate than asparagine. Okay. Which statement is true about covalent catalysis? Covalent catalysis is an enzyme catalyzed reaction. In an enzyme catalyzed reaction, in which a covalent bond is formed between the substrate and the enzyme. C is the correct answer for that. 29. Which amino acid is a strong candidate to act as a nucleophile for purposes of catalysis? Isoleucine, leucine, bad options. Tryptophan, closer to something. Methionine, our best answer is answer option D, histidine. When it comes to basically enzyme catalysis, histidine is kind of a jack of all trades. It can participate, it can act as a nucleophile, it can act as an acid. It's a game changer. The ribonuclease A mechanism is an example of acid-base catalysis that begins with one deprotonated histidine and one protonated residue. 30 is C. Now 31. The bond being broken by RNase A is A, B, phosphodiester linkage. Number 31 is B, phosphodiester linkage. Number 32, carbonic anhydrase is an enzyme which decreases the pKa of water from 14 to 7. So C is the correct answer to that. Number 33, the lysozyme mechanism revolves around aspartic acid and glutamic acid. So answer option B is our best candidate. Number 34, what type of bond does the enzyme lysozyme break? It breaks a glycosidic linkage. 34 is D. A zymogen. Zymogen is an inactive enzyme which is activated by proteolytic cleavage. 35 is B. 36, what amino acid do serine proteases have within their active site? Serine proteases. They're going to have an active site serine residue. Now, number 37. 36 with D. 37 is serine protease catalyzed reaction. Or example of covalent catalysis. Because remember that serine establishes the covalent bond with the substrate. 37 is C. Prime numbered carbons in nucleic acids are reserved for carbons within the sugar. Answer option A is our best for number 38. Number 39, I gave this one to everyone. A molecule which is 14% G is, well, it's 14% G. So D is the correct answer. Number 40, what type of bond connects the two sugars of a nucleic acid? And this is again, two sugars of a nucleic acid, C, a phosphodiester bond. When digesting double-stranded DNA, what are the two different cut patterns that can be produced? Well, blunt ends and sticky ends. The sticky ends have that 5' overhang, then the blunt ends do not. So 41 is D. Number 42, DNA sequencing is similar to PCR. The major difference is sequencings involve ddNTPs in addition to dNTPs. So answer option A is our best answer option there. 42 is A. Number 43, what is the correct sequence of cycle temperatures common in PCR? Well, we go from 95 to 55, then 72. There's some wiggle room within those temperatures, but that's going to be our best option, A, or that's our sequence. Number 44. Which amino acid is likely to be found within a transmembrane domain of the protein? Is least likely, sorry. So leucine, isoleucine, those are both non-polar residues. Arginine is a polar residue. Tryptophan, tyrosine, those are both relatively non-polar. Of all of those, arginine is the least likely. Number 44 is B. Number 45, which fatty acid has the highest melting point? Okay, so we've got, well, highest melting point, what we're looking for is a fatty acid that has no double bonds. So that leaves us A and B. And the longer of them, A is going to be the fatty acid that has a lot of carbons but no double bonds. So A is our correct answer. Number 46, E. coli grown at low temperatures will have a higher percentage of conjugated fatty acids, dispustated fatty acids, abraxas, saturated and unsaturated fatty acids. We're going to have a higher percentage of unsaturated fatty acids compared to E. coli grown at a higher temperature. So 46 is E. Number 47. Here what we got is a 14 carbon long fatty acid with an N-5. That means we've got a double bond found in two different locations, or two double bonds found in different locations. So our numbering scheme, we're starting from the methyl end. So that's number 14, 13, 12, 11, and 10. So carbon number 10. Well, when we go from the other direction, what we're looking for is carbon number 9. So that's a double bond between carbons number 9 and 10. And the other one is going to be between, remember, it's a double bond, single, single, double. So it's going to be ultimately at C6 and C9. So number 47, the correct answer is B. An antiporter is a transport protein that moves two molecules in opposite directions. Number 48 is A. Number 49, how many water molecules does an aquaporin move across a membrane at a time? 1. C is the correct answer for that. Number 50, how many sodium ions are moved across the membrane by the Na plus K plus ATPase? 3 sodium, 2, sorry, 3 sodium, and 2 potassium. And sodium are moved out, potassium are moved in. So number 50, A is the correct answer. All right, well, I hope that was helpful for everyone. And if you got any questions about this, let me know.